So I'd like to begin by, I think what we all probably, if you took any biology, what everyone sort of was taught, myself included. We really are who we are because of our genes, our genetic makeup. And really that takes us back to our mother and our father, and maybe Mendel, and pea plant crosses, and so on. And what I hope I can at least do today is convince you that maybe there's more than just genes alone. But certainly if you follow, I think everyone will know that it was roughly 10 years ago that this tour de force, the first draft of the full sequence of the human genome was published in two very outstanding journals that are in our fields. And the excitement over this was enormous. And I think it really is justified, and it really is based on what we really expected to get out of this investment. And a lot of money went into this, and certainly a lot of time and effort. And that was that really once we knew every word of our DNA, literally every base, every letter, we would ultimately understand what genes really determine disease, what genes determine aging. We better solve that quickly for me. And ultimately, we might be able to literally mine the genome for diagnostic tools, therapies, biomarkers that would really impact on all of our health and well-being. But there were some puzzles. As soon as this genome was published, there were questions that just didn't add up. They were something you really wanted to sort of sweep under the rug, but you just couldn't. And it was like, this may look like large numbers, but this was way smaller number of genes than we expected. We were really thinking there'd be hundreds of thousands. But even if you think 25,000 is a pretty big number, it was really disturbing that there weren't that many more genes in us than worms and fish and so on. And in fact, plants even surpass us. So that seems very counterintuitive, I think, to all of us. After all, we're humans. And then, of course, we're all different, but why, for example, are monozygotic twins, identical twins, where the genetic material is matched? Why can they be different? Where does that variation come from? And of course, we are who we are in part because of our past, how we've been reared. So how does that impact our genetic material? DNA is pretty fixed. DNA can't really be that plastic. So what happens there? And of course, ultimately, if you could sequence every individual who was healthy versus someone with a disease, it would be fantastic if you could literally correlate with a disease, a mutated gene, let's say. But I think I can say that's really been the exception. There are cases, but it's been the exception. Now, if we fast forward to just a year ago, what is it about this revolutionary new science of epigenetics, not genetics, that made the cover of Time magazine? So even if you really don't care about epigenetics, if you get Time magazine, you might have seen this. And they said, there's something beyond the DNA alone. And even Jim Watson said, and he can pull this off. Of course, some of you know DNA is the ultimate genetic material. It was proven to be the case here at Rockefeller University by Mac McCarty and colleagues. But Watson and Crick taught us the structure of the double helix. And Watson said there may be something more than genes alone, and that's where the excitement is now. So what really is epigenetics, and what makes it so exciting? And if you follow this field, there are some useful ways to think about epigenetics because it is confusing. Some people say, well, if the DNA is the words, then really epigenetics is how we read the words. And we all can understand that we can take a word in English and we can bold it, we can underline it, we can italicize it, we can put a spin on it without changing a letter. That might be epigenetics. Or some people have said, no. Think of the genes as the hardware of your computer. That's pretty constant. You buy a computer, you get the hardware, but it's what software programs you load into that computer that are going to tell what kind of program, what kind of output, and so on, and that's epigenetics. And what's made it exciting, the second question, is really now what epigenetics is starting to explain. I won't be able to give you all the answers, but it's beginning to connect to many different puzzle pieces. So let's just mention a few. It may well impact how we develop. As I mentioned, it may be part of what makes us different from one another. Even the cells of our body, skin, hair, kidney, brain, that may be really, again, if the DNA doesn't change, maybe this is the epigenome that's changing. We can skip down to what Mark mentioned. Are genes active or are they repressed? What's the switch? If the DNA doesn't change, what is the switch? Of course, we live in a world, we can't change that has a lot of insults, a lot of environmental change. What happens here? Maybe that's some kind of a signal that's above the DNA. And lastly, what diseases might have an epigenetic component? This works for me. If you took the actual term epigenetics, the Greek prefix epi for above, and thought about buying one cake and say, I can do that, then this may be how to think about the epigenome. Literally just add different frostings to the same cake. We do add the frosting on top and literally get a different taste for the same investment of the cake. So that works for me. This is, I'm sure, insulting to some of you. So what about a young embryo? Sperm meets egg. We begin to have a few cell divisions. But again, whatever that genetic component is, all the cells of a young embryo, they're equivalent. But ultimately, you're going to get, ultimately in development, blood cells, nerve cells, muscle cells, skin cells, all without changing the DNA. So we've known for a long time there's something else going on. The same genome, different readouts. What is the epigenome all about? And ultimately, whatever these instructions are, sadly perhaps, mistakes can be made, and if these cells somehow go backwards and sort of lose their epigenetic signatures, we may actually be de-differentiating or taking these cells backwards to a more proliferative, a more cancerous, transformed phenotype. And of course, that's not a healthy situation. So my own laboratory has been struggling with this problem for a while, and it takes us to DNA, of course. I don't ever want to minimize the DNA molecule. All of this DNA, however, roughly 2 meters in length in all of us, has to somehow package into the nucleus of every cell that we can't even see without a microscope. So it's a tremendous compaction. Estimates are 10,000-fold. And it looks like a small group of proteins, we happen to call them histone proteins, have the job of packaging the DNA. And so you might see here there's a unit. There's literally what we call in the field a chromatin unit, that's histone protein plus DNA, and it will package into what looks like a bead on a string. And then somehow this bead on a string will ultimately compress and perhaps fold and be literally shrunk down and compressed enough to be inside the nucleus. And it turns out that literally these proteins are also almost conserved in all of us. They clearly are important. Now, you might see that the artist here on the histone proteins has a few tails. You might see some tails that are wagging in the breeze. I'll actually now show you that better in a movie. So I had the privilege of getting this movie recently from a colleague of mine. Here, of course, is now I don't usually show movies, so let me stop it. Now the problem is whether I can ever get this going again. You just saw the blue DNA molecule, and here comes the first, literally, load of histones. So those are these sort of brownish colored proteins. There happens to be 8 of them total. That's irrelevant. And here's these little tails. So the DNA molecule, watch the histones come in, and they're going to literally form the first bead on a string. Here's a histone unit. It's going to wrap roughly 2 turns of the DNA. It happens to be on the outside. I'd stop the movie for you, but I don't want to do that. Now, pretend that you're going to literally have to deal with this structure. Here comes a second. Now here's the bead on a string unit. And now literally, I think I'll try to, yeah, I won't. And upon higher and higher and higher folding, this literally is what really our cells deal with. And pretend that you were called upon to switch on a gene. How would you deal with that compressed, compacted unit? And what really are the switches? So I'll take you back to the same slide you saw before. Let's start with the most decompressed, open, what would be like a slinky toy that's literally pulling those units apart? Well, it turns out the secret is in the tails. There are chemical units that are literally added to those tails. And when those tails get the sort of green ball, chemical mark, chemical tag, that actually helps to make this chromatin open up. In contrast, when you take the most condensed, compacted, you saw it in the movie, it's literally a different type of signal, a different chemical tag that's literally on those tails. Switches to red, I'll follow that convention in my talk. Green chemical units to on, off red. And literally, I think my lab is best known for literally the enzymes that we and others discovered that I like to call write these marks. And what I mean by that is they literally have the dedicated job of adding to the tails of the histone proteins. You saw them in the movie. In this particular example, I'm making it red. That would make it a silencing mark. So these enzymes literally write some of the potential inactivating marks, or remarkably, there are enzymes that literally erase the marks that take these chemical groups off. They actually peel them off the tails. So what an attractive switch. I actually don't think you could make, in a way, a more elegant switch. And of course, the logic would apply that this doesn't have to be an enzyme writer or an enzyme eraser that puts on an inactivating mark. You could actually have another system that puts on the green. But I just don't want to show the on activating mark, but I just don't want to complicate it too much. So do realize that there are dedicated enzyme systems that oppose one another that can either write or erase these chemical marks. Now here's a remarkable biological example. These images, be it fly eyes or petunia flower colors, literally in these 2 genetic situations, red here, white here, the genes are identical. And literally, what is changing the red eye color or to the white eye color is literally this system of epigenetic marks. And we know that the gene is not changing because all that's done between the left fly and the right fly is the position of the eye color gene is being moved. That's all that's happening. And the same situation applies in the plants. And I hope you're following this system of signaling to then understand that in the fly example, in the red eye state, the eye color gene has in the form of those histone proteins picked up the green marks. In the white case, that gene has just been moved to another position where it's now picked up the inactivating marks, and so on and so forth. And this pink color happens to be a complication where the marks actually get confused. But importantly, no genetic change. And yet you get this spectacularly different phenotype. I mean, literally. Now, it becomes more relevant. This is supposed to be parenting. I actually really like this cartoon. I think this is parenting, right? And I had to only add epi. The artist didn't put in genome. This wasn't when it was cool to write about epigenomes. But you can see the mom and dad are hard at work, and brother and sister, of course, are having some trouble. And actually, she's saying it just right. She's really probably bragging about her epigenome being, more complicated. He's upset about it, but she's actually got it right. So let me give you an example of why that's true. Of course, we all know that the men, and this would include the father, we're XY. In the women, the girls, they're XX. It holds for the mom as well. And that imbalance of having an X chromosome but only one in the male and two X chromosomes in the female is not acceptable. That's an imbalance. So I think it's fantastic that in women, and only in women, one of the X chromosomes has been randomly inactivated. So again, identical genetic material, but one is fully off. And I'm showing you that as the little xi. And in other cells, it's the other X chromosome that's randomly shut off. And again, you might be guessing, indeed, this picks up the inactive, what we call the inactive X chromosome, picks up the silencing mark. And I'll just show you that we can literally take female cells in the lab and stain the cells. I'm giving you the outline of the nucleus, and this sort of orange blob is literally an antibody that has been developed against one of these silencing marks that is literally staining that entire inactive X chromosome. If you did, in females, if you did this same experiment with male cells, you would see nothing. If you had a triple X situation, you would see two of these. And as I think an interesting footnote of biology, this is why calico cats are always female. This is tied to this random X inactivation that's going on in a way that dictates the spotting that makes every calico cat literally a female. So we're back to the puzzle that I showed you earlier, and I thought it would be most interesting to skip right down, I don't want to go too long, to what about environment, and then ultimately what about disease. And I think this is one of the most amazing single figures that I've ever seen. So let me take you through this. These mice have been arranged from left to right. You can see a very different coat color. It's an agouti tan color. They're called agouti mice. But really the genetics is tied to a problem that literally causes these mice to overeat. They become fat, they become diabetic, and they die young. And of course you can see that there's a nice progression. We're at the other end of the spectrum. We've got a much smaller mouse, different coat color, healthier weight, live longer. What's remarkable about these mice is not that they're the mothers, they were actually the pups. I have to tell you that these are the kids from mothers that were literally fed, as pregnant mothers, a vitamin that literally we know now is the donor for these inactivating, kind of, we'll call them red, chemical marks that are put down on those histone and DNA tails. That's just remarkable. So the mothers were fed different diets with different vitamin supplements, and this is what their children did. And the children here are all genetically identical. I think that's just a remarkable, and in this paper published in 2007, these authors concluded with this sentence: epigenetics provides a plausible link between environment and alterations in gene expression that might lead to disease. Now, 2007 is a while ago, but it's not that long ago. And the very same authors, I just think you'll find this interesting, sponsored a meeting not that long ago, this is last month, literally, where this was the title of the meeting, Environmental Epigenetics and Disease. I didn't want to lift all of the topics. It's just too hard to read. But let's just bounce through some of them. The environment might impact on cancer, cardiovascular disease, schizophrenia, female health reproductive events or health effects, depression, psychosis, early life impacting on the epigenome, and autism. And certainly autism needs no introduction. And this is the cover of what was the first ever dedicated Cold Spring Harbor symposium that was literally dedicated to the topic of epigenetics. I think it's a very provocative cover. They obviously keep this very, very confidential, but these are two identical twin sisters. You can find out if you talk to the folks that put this together at Cold Spring Harbor, one of these young women is completely phenotypically normal. The other is actually a very poorly functioning autistic identical twin. And of course, this is really one of these puzzles. Individual variation with an identical genotype. And another, I think, startling aspect of the autism spectral disorders is most of these kids start out normal. Their first year of life, things look completely good. That's true for the two women on the previous slide. But as they progress in age, things start to sort of happen. They start to have really a loss of ability to speak well, social interactions, seizures, stagnation, and then all of a sudden, hyper anxiety, motor deterioration, and in their late 20s, teens, they're beginning to pick up Parkinson's features. And again, here there's a time variable, but there's not a change in the genotype. There's not a change in the genetics. So what's going on here? And I just think from a very new, it was something that I got to watch this weekend, a nice new report from Robert McNeil, a PBS special that was called Autism Now. They cite in that piece that 5 years ago, autism was being sort of ticked out at a rate or a prevalence of about 1 in 3,000. Now it's roughly nationally at 1 to 100. And some friends of ours said that actually New Jersey is at 1 to 50 on a national average. And I think those are scary numbers for all of us. That's really quite a change. Sure, we're diagnosing it better. Sure, it's being looked at more carefully. But I think those are pretty staggering numbers. And I'll just sort of leave you, because we want to leave a lot of time for questions, with one last tidbit about this sort of chromatin layer, is we're just now beginning to find out that in my own lab, this is very recent new data, that not only can these chemical tags change, be it active here or inactive, but you even can swap out the histone protein itself. So I'm trying to show here a different colored histone, and with some dedicated machinery, you can literally like reshuffle the deck and get the yellow histone in and the previous other histone out. And now it looks like this is a very, very, very potent mechanism, especially in the brain, for changing the sort of epigenome in a way that might correlate with, hopefully normal development, but again, mistakes might be made. And I just want to comment that my own laboratory this past year identified with this particular histone variant who are the components that shuffle in this one particular histone variant. And 2 proteins came up. One is already very closely aligned with a mental retardation disorder that makes us wonder about sort of what's going on in autism over time. We raised an antibody to this variant, and literally as the brain gets older and older and older, there's more and more and more of this protein building up in the brain. And we're now starting to find that even when we manipulate the mice with learning and other kind of enriched environments, there's more and more of this taken up. So we're very excited about this. But never in my wildest dreams would I have expected this, which is really why I'm in science. The proteins that literally put in this histone variant were just identified not but 3 months ago with a tour de force study that came from Johns Hopkins Medical School in Baltimore, where the cancer docs there were literally looking for genetic mutations that correlated with a rare form of pancreatic cancer. It is the pancreatic cancer that Steve Jobs has, so that puts a face on an individual that suffers from this disease. The good news is that this isn't one of the pancreatic cancers that is literally such a killer so rapidly, but it's absolutely remarkable that it's in the machinery. The mutations in this rare form of pancreatic cancer are in the machinery that we identified that puts this histone into the chromatin in this particular situation. So we're suddenly really left with a very provocative kind of puzzle, which is good for science. What is it about resculpturing the epigenome in potentially neuronal tissue, but somehow some kind of a link to mistakes made in a particular subtype of pancreatic cells that correlates with the rare form of pancreatic cancer that Jobs has. So I think we have our work cut out for us, but that's really a remarkable, I think, coincidence. And of course, at the end of the day, do any of these identified molecules provide useful drug targets? This is the first example that I'm aware of that was actually developed against drugging one of the drug targets. Remember when I had my slide on writers and erasers? This is literally what was drugged in this situation. It actually was a drug that was developed at Memorial Sloan Kettering. It ultimately was the first FDA-approved therapeutic drug that sort of fell into the epigenetic-based drug category. It's now been handled by Merck, but here's an individual on the left-hand side before any drug treatment was made. This is two scans of a chest cavity from a metastatic laryngeal cancer that spread to the individual's lungs, and after treatment with this epigenetic-based inhibitor, this is the same individual 8 weeks later. And I'm happy to say this individual is still doing well. So at the end of the day, we can sort of plot what's happened to the citations on just the word epigenetics in the title or the abstract of the formal literature. And so you can see that it's really, really ramping up quite a bit. But even if you don't care about papers, there's an awful lot of proteins that now seem to be dedicated to resculpturing the epigenome. Many of these proteins and enzymes have now been drugged. Their structures have been solved, they're being drugged, and these drugs are now in active clinical trials in this country and beyond. Many biotech companies have now recognized this may be a very attractive space, and it's moving into, as I said, big pharmaceutical programs as well. I want to pay Rockefeller some credit. Epigenetics is now sort of being dubbed as a new science. Sort of that's true. I showed you the graph of how scientists are studying it in terms of the literature, but this is actually Vince Alfre, who spent his entire research career here at Rockefeller University. He was committed, and he is often given, I think rightfully so, credit for being the person that said the chemical marks, which I've neglected to tell you what they are, the red balls and the green balls are acetylation or methylation marks. He is largely given the credit for being the person who said, these are going to be important, these are going to be the molecular switches that govern gene regulation. And sadly, I didn't get to be here at this campus when he was alive, but I think he deserves much of the credit for being way ahead of his time. I want to make sure to thank all the folks in my own lab. You know, if I showed you anything from my lab, recognize that I didn't do it. And we've had always a nice aspect of being in this university, in this neighborhood, is just how much great science is done here. And we certainly enjoy collaborations with many labs. I wouldn't have moved here. I was literally convinced, and I won't say who, but somebody told me, you know, if you come to Rockefeller, it's scientific heaven. And I really have now recognized 8 years into being here just how true that is. This is a very special place to do science. And I'm very grateful to all of the individuals who make Rockefeller happen. And of course, thank you for all your time and attention. I don't want you to applause yet because I really worked hard on one thing that I realized I didn't go too badly over. I'm going to get in trouble with Marnie for this, but I'm going to skip a blank slide. Now here's another reason it's no more data. I'm done, really. Mark has never even heard me talk, so this is probably, I'll be looking for a new job tomorrow. But another thing I like about science is the talented people, and of course the graduate students here are phenomenal. One that was here, he's now an assistant professor at Hopkins. He was absolutely a phenomenal cartoonist. And one of the cartoons he drew for me, I think, is actually relevant. Sean Taverna, I should say. This is Sean. Again, if his scientific career doesn't work out, I think this is going to be his next Gary Larson phase. But the DNA molecule is literally stepping out of the shower and has to make a choice. I apologize to the women here. I don't know who chose the Histones versus the Herstones. But I do want you to recognize then that there is another layer, and this layer is being regulated in an elaborate way. It's a staggering amount of complexity, and it seemed appropriate to me to show you this cartoon, to show you what Sean's leaving present was for me. And just one more click. And now, how could I not show you? Maybe some of you know where this is going. If I don't hold you up here tonight, now, there is one science part to this. In the latest issue of Cell, that's one of our premier journals, they have dedicated the first page or two to the royal wedding. I want you to know there's a royal way to package our genome, and I hope things work out very well for Kate and William. But they say in this chromatin article, epigenetic changes can transform a commoner into a queen. And what they're referring to is all of the genetically identical bees, when they hatch, it's the first bee that gets the royal jelly that's the queen. So think about what is going on when all the genetically identical offspring, one gets to the royal jelly, she's the queen. There you go. Okay, so.
First, David, I'd like to thank you for a fascinating lecture. And there's something I'd like to bring to the audience's attention, if I can have the slide. There we go. David was much too modest to mention that he is one of the key players in this field of epigenetics. And I'd like to illustrate it with this slide, which you saw earlier. He did show you this slide. There's this dramatic upswing, as you've seen here around 2000. What David didn't mention is that this inflection point was triggered by a very important event, which was the publication of David's paper on the histone code that year, and which sparked this gold rush and an amazing amount of interest in this area of study. I'm sorry, David, if I'm embarrassing you. I didn't tell him that I was going to put this up here, but I think that it's important for you as the audience to know what a key figure he is in this field. So in the remaining time, we'd like to take questions from the audience.
That was an excellent discussion and representation of some amazing research. I would like to ask whether or not you've identified the 2 or 3 things that, as parents, we should be aware of that is environmentally focused that could affect the epigenetic content of our children?
Well, that's obviously a very important question for parents, and I think we're really early on that curve. Not this curve. We're really early on this. I think there's been some phenomenal studies. I showed you the mouse diet picture, so I might say diet. And now there's been published, I think some of you may have received the Time cover that I showed. Some of you might have seen that Time article. But I think another aspect is sort of our lifestyle. And you would have said, sure, if we're going to take a bullet, hey, that's our choice. But in now some amazing studies, it's not just what you're doing. You're going to ultimately maybe pass this on to not only your kids, but your grandkids. So, I mean, I think one aspect I'd say would be the diet aspect. Another aspect would be the lifestyle. And, you know, we would have been paying, I think all of you would be paying super careful attention to the rearing and, you know, the nurturing. Bruce McEwen's here, and one of, I think, his former PhD students from Rockefeller, Michael Meany, is now showing that with mice, if there's good moms, we'll say good moms, they're heavily licking and taking care of their babies, versus bad moms who really kind of just go off to the other side of the cage and neglect, that's causing literally disease problems in the animal, but most remarkable, that's being propagated to the next generation down. And so, I mean, these are just unbelievable studies that are early in the curve. He's in McGill University, and now I think Canada is very actively now doing this very kind of sort of study. I'm not actually sure how they're identifying good moms and bad moms, but they're looking now at kids who are coming out of these two very contrasting environments and looking at their epigenome. And startling. So those would be some things that I think we can all relate to, you know.
Other questions? Here, down the front row.
So with that information, how do I correlate that to the information you showed us on autism, particular to the twins, for example, where you have two people? I'm assuming the mom is not treating them that differently.
No,
so
that is, of course, the issue, I think. And there's, again, a very wonderful, sort of new, at least it was new to me. My wife turned me on to it, a Nova special that was called Autism Now, that Robert McNeil covered because one of his grandchildren is autistic. But I think I won't be able, I think, standing here to now sort of in any way tell you, how what I talked about molecularly right now can sort of really be brought to bear on improving the situation. Of course, autism disorders are extremely complicated disorders, and I think we're excited that maybe some of what we're just now starting to do in my own lab with the brain work will help move some of that forward. But I don't think I'll be able to give you a go out and do this. I really won't. I think that would be way out of line for me.
There's a question here.
So bad answer.
First, thank you very much. I think parents today, we have a sense that we can control a lot, and this confirms in the environment with our choices and chemicals, etc., lifestyle. The suggestion, though, also, is it possible then that making changes at the behavioral, psychological level can ultimately also impact markers at the epigenetic level?
Well, it's a good question. I think that, in as much as sort of like the Michael Meany studies that I mentioned, where it's mothers being good to their newborn pups versus not so good, that really, I think, speaks to behavioral, changes that I think ultimately labs like Meany and others will ultimately get a handle on. I think it's even exciting. Mary Jean Creek's in the audience, but her laboratory is focused on chemical addiction. Now I think people are beginning to even find out that, addicts, whatever the problem is, alcohol, hard drugs, that even individuals that now have sort of gone down that route and want to sort of kind of get back on track, they are ultimately going to have to sort of reverse some of these imprints. And there's been some remarkable studies just last year that, the writers and the erasers of the silencing mark, the red mark, they actually now have been drugged well enough that not only are they being used in cancer trials, but they've actually been used to see if they can reverse cocaine-induced behavior in mice. And giving cocaine, hooked mice these inhibitors of the epigenome literally can reverse their addictive behaviors in mice. So that doesn't really get at behavior except bad behavior or a problem. And Mary Jean's here. Her lab is actively looking at that as well. It's fascinating.
We have 2 questions up there, back side by side.
What determines the pattern of acetylation and methylation?
A good question. So, I certainly just said there's a tail. I said you put on the mark, but it absolutely is so precise that it is literally in the writers and the erasers that choice is being made. And, in a human, we have, depending on whether it was the green signal or the red, the ballpark of how many enzymes are doing that job is literally from 50 to 200 of each class. And frankly, every time we go into the cold room and purify another complex biochemically, we get something that's a gene product that's often associated with mental retardation. I think the brain is going to be the exquisite place where the epigenome, because think about it, you've got all this like neuronal demands for cell type specificity, wiring. I'm not trying to speak to my boss, but in one very new result, we're talking this last week, we used an antibody against that histone variant, and we asked, where does it sit down the genome? Now we're back to the genome, the Watson-Crick DNA. And literally the top class of genes that associated with that variant were literally the axonal guidance signaling pathway that Mark helped break open in his lab's work. So then, why am I excited to be at Rockefeller? I can bug him now and his lab members, maybe more his lab members. But anyway, a staggering complexity of gene products that are dedicated to that specificity of which acetyl, which methyl, which chemical mark.
We have a question. We've moved down. Sorry, we'll come back to you.
Apologize in advance, I'm not a scientist. But the markers, if they're changed by enzymes, switched on and off,
correct,
is each enzyme unique to each?
They're quite unique. It's not an absolute.
Run the risk of, say, turning the fridge off at the same time as turning the lights off or something.
It's not as simple as sort of one mark on. I obviously made this, hopefully, not trying to overbear you with, too many marks, too many locations of the marks. But I would say that in general, especially with the red silencing mark, when you put that on a particular key spot on the tail, that gene will turn off like no tomorrow. And again, if you literally haven't changed the DNA, if you can get a drug, get a therapy to get that red chemical mark off, that gene's going to reactivate. And if that's a gene that's protective, like good for us, tumor suppressor, good for us, that's a wonderful therapy. And I should say, I said 180 some clinical trials. In some particular forms of cancer, the success rate of these drugs is over 50% in patients that thought it was, 6 months. I don't think it gets much better than that. That's maybe a very limited cancer type. But for those patients at the individual level, that is really impressive. So the more these enzymes get drugged and put in different combinations, probably we're going to see some even more I hope, some more remarkable therapeutic good outcomes.
Maybe we could, the person who is overlooked back up there, and then we'll come over to the center, this gentleman here. And
Thank you. As a parent, a question regarding the identical twins as well. Birth order, birth process, health of the mother during the pregnancy, are all of those holistic ideas brought into the discussion about?
Wow. I think certainly the reproductive health aspects for sure. It's getting attention in the field, in the broadest sense. Birth order, that I can't think of off the top of my head study that I'm aware of where someone has now. I mean, we kind of have paired with. I have 3 children too, like Mark, and we've kind of talked to enough other parents that have, like, wow, is your first kid like our first kid? So I mean, there are things like that that. I think it would be fascinating. I think your question is actually quite good because there may well be some kind of an order, and if that had its, an epigenetic connection, it's kind of like that first bee that gets to the royal honey. That may be something that I'm just not aware that that's been looked at. But I wouldn't rule out the epigenome. I mean, think about the possibilities here. If I can tell you there's roughly 50 to 100 writers, 50 to 100 erasers. I didn't want to bore you with lots of different chemical tags. I mean, I think there's very little options for the DNA. There's an awful lot of options for the epigenome layer, the frosting.
gentlemen.
Turn it on? No, it's on. Have you considered people's belief systems and thought forms in epigenetics? I read the book by Dr. Bruce Lipton, Biology of Belief. If you're familiar, are you familiar with it? He's a cellular biologist.
Okay.
I think it was a New York Times bestseller about 6 or 7 years ago, Dr. Bruce Lipton.
Okay.
Where he, as a cellular biologist, has proven that our belief systems and thought forms are literally in our DNA, and that has a huge role in epigenetics.
Fascinating topic.
No, you're not familiar with it?
No, I'm not. I'm not. The book, or I mean,
The book.
I'm just not.
I'm a energy healer, so in the world of vibrational medicine, I mean, he's very well known and
I don't
respected.
dispute that at all. That's my ignorance, not.
Check it out,
I
because
will.
it may have a huge role in this.
The deficiency is all right here. I promise.
No, it's just an indication of how much there is out there to learn
Oh,
from.
absolutely. And the graph sort of says, the graph I showed, I mean, really, this was so curious, like, we'll go back to sort of to the 2000 mark, 10 years ago, really, why were these identical genes being sort of silenced when they were moved in a genome? People were like, this is nutty. And now it's becoming like really, really, really actively studied, biochemically, I think therapeutically and so on. And so I think part of I think my mission is to try to let people know there is something beyond the DNA. Realize anytime I said there was a mutation, or the cancer folks that sequenced the pancreatic cancer, when I say they found a mutation, we're back to real genetics. We're back to, but it's interesting to us that then some of those mutations map to these epigenetic regulators. So I think, if you think about it sort of like the icing on the cake, once you've made mistakes in that icing, it may cause problems. And of course, a lot of you follow, I'm sure, stem cells, stem cell reprogramming, the therapeutic options. I mean, what it looks like is to really take a liver cell or a skin cell and actually to ask it to regenerate, parts of the body that have maybe been damaged or destroyed. I mean, you have to get that backwards into like a more embryonic state. And people now think that the epigenome is sort of a layer that has to be dealt with. It'd be nice to be able to erase the epigenome, wipe off that icing, and then redecorate the cake. And I think that, some of what you're referring to that I'm not at all on top of, may impact on that. Interesting.
Gentleman here.
How are you given the complexity of both the genome and the epigenome layer, how are you making your choices of what to be looking at and studying and applying your lab's efforts to?
That is certainly hard. I would say that we exploit as many different types of biology as we can. So, for example, when I showed the inactive X chromosome as an example of two identical chromosomes that in female cells, I think that literally that is so powerful for us. We look for model organisms where there may be a lot of the inactive chromatin. And then if we purify that and then we sort of really subject it to detailed analyses, sometimes a difference in a mark just jumps out. That certainly happened for us in some of the cases that we've already really actively pursued. And then the trail for us is pretty easy, honestly. We pick that mark. We literally develop often an antibody. That enables us to sort of explore its biology. And then we just go to the cold room and say, can we purify who writes that mark? Can we purify who erases that mark? That's a lot of work. I take no credit for most of that. But then for us, a lot of times that's proven to be very, very useful. But I think on the other hand, if you said, have you just touched the beginning? Is this the tip of the iceberg? I often feel that. We're probably just scratching the surface. Part of it is, how many marks are abundant enough for us to see? So it's a great question. Have we guessed right? Have we picked all the right marks? I think we, meaning the field, have begun to crack this open, but by no means do I think we're done. And if I picked a mark, well, pick your favorite mark, whatever, red or green, think about the combinatorial aspect. What if you linked up a green with a red and a green, all down one tail, one histone tail? What would that signal be? As opposed to red, green. Who wins out? And so, it's staggering complexity. But I think, when I said maybe the Genome Project didn't give us enough genes, I think the sky's the limit on the epigenome.
I'm not sure if I understood this correctly, but I thought that you said that some of these marks could tell the DNA to move something down the genome?
Oh, I didn't say that well. No, so I was confused. When I showed the fly eyes, and one was absolutely red, one was absolutely white, the only difference between those red versus white eyes was the position of the eye color gene on the chromosome. So let's say in one fly it was here. Well, let's say that was the red. Then it was on, the gene was on, it made red eyes, and it picked up all those green signals. But if you just do the experiment, literally, of take that single gene and molecularly move it over here, now suddenly the eyes are off. The gene color is off. It's white eyes, and now you isolate that gene and study what marks are on it. It's the red. So green over here, on, over here. And so it's literally us molecularly moving the gene, literally, to a new position.
One other question on a different topic.
Okay.
It sounds as though behavior could affect the epigenome because it creates enzymes in response to something like the enzyme that's created when you go running or that sort of thing. So does that mean that different cultures which encourage different kinds of behavior could be affecting the epigenome of that entire culture?
I would certainly not rule that out. I think, the Times article that some of you may or may not have seen was a very small population of Swedes that were so far north in Sweden, if they hit a famine problem, it was sort of their problem. And what these researchers that now are in Stockholm and literally did that was phenomenal is they followed these families because the records were so good on the crops and the children and the grandchildren, and it absolutely tracked, their diets. But not just the ones that sort of, let's say, were generation one, it was the past parent generation, and they could track to when the famine came through, if it was the young, let's say, 20-year-old male, and then, it wasn't a problem for them, let's say, they survived it, but down the road, their kids had trouble, their grandkids had trouble. It's unbelievable. So I don't think that's a cultural thing, except just their isolation. And then it dictated to famine or feast. And when the times were good, the children and grandchildren had different medical problems. So if a culture has a certain sort of style, I think it might well affect the epigenome.
Well, that's all the time that we have for questions here, unfortunately. But I'd like to invite you to the reception in the lounge upstairs, where you can ask David any additional questions you might have. And I'd like to thank him for sharing us his insights. And I'd like to thank you all, also all of you in the audience, for coming out to participate in this evening's program. Thank you very much.